Hey all, so this is the week 3 lab cell structure and function, cell types and transport video walkthrough. So as always, we're going to use that lab manual, read it through completely before you do it. You're going to complete the document and then everything goes into that document. You want to see the faculty materials because those are mine. You get the formatted lab, the rubric, and the playlist, which you're probably using now to watch this. So let me pause this and pull up that rubric first. So here's the rubric. So you see we've got observation tables again, a point for each exercise, half a point for grammar, and then as always put it all into there. Let's go look at that lab report. Okay, so as always, title page first. So your title, name, course number, date, and my name is optional. Let me scroll down, blah, 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 back to observation. So you're going to fill in the organism name for your prokaryotic structure, and then you'll do it for your eukaryotic cell too. And then we're going to include our drawings down here. They can take more than one page if they need to, but make sure you include those drawings. And then we're going to have what's our medium, when did it start. You can just say 0 and then pick your end time, how many minutes, or you can say 1 o'clock, 2 o'clock. So 1 o'clock to 2 o'clock would be 1 hour. And then how much it moved, what's the diffusion rate, which would be per an hour. So make sure this is in hours. And then what's in your dialysis tubing, what's in the beaker, what's its initial color, final color, what did Benedict say the color was at the second part, and so what does that mean is in the final thing, right? So you're guessing if this says nothing, then whatever this tests for probably moved over to here, right? You know, I should actually probably pull up lab answer key, which I haven't actually done yet. So let me do that. Apparently I'm just I know all of this. I know it all. I'm amazing. This is the really long tile. Okay, really long tile. So exercise 1 is prokaryotes and eukaryotes. So we're going to focus on that, the differences of cell structure, right? Blue, blue, blue. I've got it here. Scrolling. All right. Boop. So what are the differences that you see in the two cells that you drew? What are the specific cell part that each cell type possesses? Say 2? That sounds weird, isn't it? Oh, that's because there's answer below. Hello helps awake. So to do what? So escape hazardous conditions, you put whatever your answer is in, regulate water content, store food. This one is not going to be the cell part, it's going to be which of the following does it do. Okay, does that look weird? It does. That's why. Center. Let's check this one. Always the tabbing. And as always, if these little things bug you, hit the paragraph symbol. They go away like magic. But they make me happier when I'm editing. Okay, so questions. With which cell type are you most familiar? Can be either one. I'd say why though. Whatever, because I see it all the time. Whatever, because I've studied it. I don't know. Which cell type is more successful in an extreme environments? Why? Because prokaryote to bacteria are generally 3 micros or less in length. They may not exhibit specific structures under a compound microscope. What type of microscope would be the best choice for viewing the bacteria? So if it's really tiny, which one do you want to be using? This one looks wrong, doesn't it? That's right, this one's wrong. See? Seriously, spent all this time and they're still off. Okay, so research, that means in-text citation, full reference at the end of the report. Amoebas online, and give a few examples, including where they're found and what energy source they are. A few examples, that means specific. Maybe we should just put a few specific examples. Specific examples. So give me really specific, like a genus, species. Where is it? What is it? A few examples, at least 2. Right? Discuss whether cyanobacteria and amoebas are beneficial or detrimental to humans and to the environment in general. So talk about both, right? And then decide which one is more. And then we have some really odd spacing, don't we? We'll just fix that quickly. So now we're going to talk about plants and animals. So no more prokaryotic, eukaryotic. Now we are all in use. Eukaryotics. So now what's the difference between the two cells that were drawn of our plant and animal? okay? And then we're going to talk about the same thing. What do they do? So what do they use to regulate water, energy, store food? Store food is kind of weird in prokaryotic, so be creative or ask me. It's a weird answer. And then reproduce, you're going to answer with one of the following. So instead of saying like the nucleus or something, you're going to say it's done through division, it's done through asexually, sexually, it's done through budding. You know, it's weird, but that's how they did it. So that's what we're doing. And then which type of cell, plant, or animal are you most familiar with? Why? Which type of cell would be the most successful in extreme environments? Why? And then below, you're going to give me something, the differences. And you can literally put things like, because they're different, right? So one thing might be on one side, like I might say the plant has a chloroplast, and over here saying no chloroplast. That works. And then similarities will be things that they both have. Fill in everything, even if I say like on one side it says no or no over here, and this side it has it, and then similarities, you can easily come up with enough. So then we're going to be looking at the transport of materials through our cell membranes. Let's see, this one says passive transport, right? So remember that might become later important. This is passive. So which medium had a faster rate of diffusion? Why? Then we're going to graph the two sets. Let me look and see what they did. They did a line graph with time on the bottom and the movement diameter in millimeters on the left. Okay, and then you do one for your liquid, one for your semi-solid. So were your hypotheses supported or refuted? How might the rate of diffusion be increased or decreased? I feel like we always talk about temperature. How might this be tested? So how could you test whatever you just said? Why is this exercise an example of passive transport? Why must the spread of coloring and gelatin in the gelatin be monitored? Why can't you just leave it? What would be the graphic method to detect when the rate of diffusion in the gelatin is at a maximum and when is it beginning to slow? So what might we have to graph? And then, so if we did the graph we just did, how would we be able to tell when it started to slow down? Okay, what would the graph look like? So we construct a table. This should be a table, huh? Insert table below. See, look at you guys. Okay, so you're going to have a table down here that shows what the averages was for your classmates. Okay, so then we have transporting. Now we're doing active, right? Active. Also might be important later. So what are your test substances? What are you looking at to see if they're moving around? Why did a color change occur in the jar? So like also you doing a color test, right? So that should tell you hopefully which of your test substances are. Why did the color change in the jar occur? So what does it indicate that you put in there, right? Why did the color change occur in the dialysis bag? So did something move? What does Benedict's reagent test for, and what did the test of the jar indicate? So what is it showing you? What did not diffuse? So what just hung around, didn't move, and why? Like, is it heavy? Is it big? Is it slow? Is it what? So then were your hypotheses supported or refuted? How might dialysis membrane tubing be similar to a plasma membrane? So we said something didn't cross through. Why is that like a membrane? And so what type of transport mechanism does this exercise simulate? What was the title? And what can you conclude? So does everything get through? How would an actual membrane decide on this? Right? And then our lab summary is at least 3 sentences summarizing what you learned, so all the various parts. You could have a sentence on each exercise, which would give you 4, make life easy. This one you got to have some references in here, so there's your reference section, and you put your reference in here. Make sure you have your hanging indents. If you use a website and there was no author, use the organization as the author. Hopefully you'll be really good at this by this point, and it'll be really easy. You know, this lab went through really quickly. I feel like I wasn't telling you a lot, but there wasn't too much to talk about, right? Because most of it's drawing and like all the plant and animal, eukaryote, prokaryote, that's stuff that you just kind of learned about, right? We haven't gotten to play with it yet. soon we'll get to play with it. And then you're playing with that tubing to recognize the different types of transports. So hopefully this helped. I love a draft before noon on the due day.